Um, we'll be talking about the extracellular matrix and cell-cell interaction in this lecture. So let's start with the matrix. So the extracellular matrix is the molecular composition of the space between cells. It is the molecules that connect tissues and cells to each other. And there are different types. We have what is known as the basal lamina, and we have connective tissues as well. So the basal lamina basically is it's a thin sheet-like structure where epithelial cells lie on, they sit on, they rest on. So these are the epithelial cells, and this is the basal lamina right here. So what happens, so what it does basically is that it supports sheets of cells and it separates these cells from the connective tissue and the cells that are present in the connective tissues. Now, the connective tissue is basically a loose network of proteins and carbohydrates underneath epithelial cells. So this is where you have all of the other cell types like muscle cells, fibroblasts, endothelial cells, sometimes inflammatory cells, blood vessels, and so on. Now, there are different types of connective tissues like bone, tendons, and cartilage. So there are different compositions of extracellular matrix. So we can categorize them into three types. We have matrix structural proteins, collagen and elastin. We have glycosaminoglycans, which can be associated with proteins forming proteoglycans. And we have adhesion proteins that mediate interaction between cells and the extracellular matrix, including fibronectin and laminin. Now let's start with the matrix structural proteins now. Now these are tough, fibrous proteins. Okay, they form gel-like. They can be embedded in a gel-like polysaccharide substance. And usually you have adhesion proteins that link cells with matrix, and you have adhesion proteins that connect these matrix proteins to each other. Now, the basal lamina, as an example of a structural protein, is formed from components that differ from those found in the connective tissues. So we have tendons, for example, and tendons contain high proportion of fibrous proteins. On the other hand, you have cartilage containing high concentration of polysaccharides. The reason is that, if you remember, with the glycosaminoglycans, if they are compressed, the negative charges of the glycosaminoglycans would prevent, first, cartilage would absorb shock, and then the negative charges of the glycosaminoglycans would expand the cartilage back into its relaxed normal structure. Now in bone we have the matrix, basically collagen, hardened by deposition of calcium phosphate crystals. So you can see that connective tissues have different composition from each of molecules from each other. So let's start with the collagens now. The collagens are the most abundant proteins in mammals. So they make up 25% of total protein mass. They are long, stiff, triple-stranded helical structure made of 3 alpha chains. The basic unit is known as a tropocollagen, and it is rich in glycine as well as proline and hydroxyproline. It contains hydroxylysine, and hydroxylysine is important for attaching polysaccharides to the protein, so the protein is a glycoprotein. And hydroxylysine is also important in increasing the number or the proportion of hydrogen bonding within the 3 strands, so that the collagen molecule, the tropocollagen, would be quite stable. Hydroxylysine or lysine can be oxidized, allowing for the cross-linking between the different lysine residues, or the modified lysine residues, or the aldolysine or all lysine. We already talked about collagen in basic biochemistry. So there are different types of collagen molecules and different distribution of these types. So we have the fibrillar collagens. So basically they are the long strands of collagen molecules. We have the fibril associated collagens that link collagens to each other. We have network forming collagens. We have anchoring fibrils, and there we have also transmembrane collagens as well. So there are different types of fibrillar collagens. Okay, the most common is type 1 collagen, which is present in most connective tissues. It's long strands of collagen molecules. These strands are aligned in parallel to each other, and this type 1 collagen is a rigid molecule. So it is present. It's the main component of bones. We have type 2 collagen, which is present mainly in cartilage and vitreous humor. It's smaller in diameter than type 1. It's oriented randomly in the viscous proteoglycan matrix, so it is mixed with proteoglycans. It's a rigid molecule, but it can be compressed, okay, resisting deformations in shape. It can absorb shocks as well. There is type 3 collagen, which is present in skin and lung, and it can be extended. We have type 11 collagen, which is present in cartilage, and we have other types present in different types, in different tissues. So how are collagen molecules assembled? Basically, you have the production of one strand of a collagen molecule known as the alpha collagen. 3 strands coming together forming one molecule known as tropocollagen. Now tropocollagens can associate with each other, and they can be cross-linked, by the way. So we can have this covalent cross-linking between the lysines or the oxidized lysines within the tropocollagen, or between the different collagens forming fibrils. Different fibrils coming together forming large collagen fibers. Now we have other types. So this is type 1. Other types include fibril associated collagens. So these are smaller, but they can be associated with the fibrillar collagens. And these include collagen types 9 and 12. So they link fibrils to one another and to other components of the matrix. We have network forming collagens like type 4 collagen. So you find here type 4 collagen right here, okay. And this is a component of the basal lamina, by the way. And we have anchoring fibrils as well. They link the basal lamina to the connective tissues. Okay. Now, how is collagen synthesized? Well, you have the synthesis of an alpha chain, and this chain is actually synthesized as a protein. Okay. Now in the ER, you can have modification of prolines and lysines, so that some lysines can be hydroxylated. Now one alpha chain can combine with 2 others, forming pro-collagen. Okay. Remember that here you have the formation of a tropocollagen, but still the pro regions are still there in the protein itself. Now, following exocytosis of the pro-tropocollagen, you have extracellular enzymes, including pro-collagen peptidases. These enzymes remove the pro regions from the N-terminus and the C-terminus pro-peptide, forming the mature tropocollagen. Now what happens then is that following the removal of the pro regions, now the different tropocollagens can form fibrils, and fibrils can form fibers. Okay. So this figure illustrates the synthesis. So right here we have synthesis of the pro-alpha chain in the endoplasmic reticulum. You have modification of prolines and lysines into hydroxyprolines and hydroxylysines. Then you can have in the Golgi apparatus, you can have glycosylation, glycosylation of the pro-tropocollagen. You can see the pro regions right here, the propeptides at the N and the C terminus. Then the collagen is secreted outside cells. What happens then is that you have the peptidases removing the pro regions, forming the fibrils and fibers. So basically, there are two reasons why formation of collagen fibrils and fibers does not happen inside cells. The first reason is that the propeptides inhibit fibril formation. So as long as they are part of the pro-collagen, you cannot have fibril formation. The other thing is that this cross-linking that takes place within the tropocollagen and between different tropocollagens does not take place except outside of cells, because the enzyme lysyl oxidase is localized outside of cells. So collagen is a highly cross-linked molecule in tissues, and its tensile strength is required for Achilles tendon. So if this cross-linking is inhibited, the strength and the function of collagen is not there, does not exist anymore, or it gets really reduced. So tissues become fragile and they tend to tear. In addition, collagen, any deficiency in the synthesis of collagen is related to another condition, and that is scurvy, or another disease known as scurvy. We already talked about scurvy in biochemistry, and it's related to the inability to hydroxylate proline, and due to deficiency in vitamin C. So the thing is, the lack of hydroxyproline results in weak collagen interaction, intramolecular and intermolecular interaction, that is within the same tropocollagen and between the different tropocollagen molecules. So tissues would become weak, including skin. It becomes easy to be injured. Gum, teeth would fall down, blood vessels would tear apart, and so on. Another condition related to problems with collagen is osteogenesis imperfecta, or it's known as brittle bone disease. So the thing is, osteogenesis imperfecta means imperfect bone formation. So the condition is related to easily broken bones, for example, due to weak collagen molecule. Now, there are different types, or 4 types of osteogenesis imperfecta, and they're designated as type 1 to type 4. Type 1 is the mildest, type 2 is the most severe. So there are different ranges or different severity levels of that disease. So this osteogenesis imperfecta is related to mutations in collagen 1A1 and 1A2 genes. These are the genes that are responsible for the formation of type 1 collagen. So what happens is that in osteogenesis imperfecta, due to mutations in these genes, the bones would be weak. And it is an autosomal dominant pattern disease, meaning that only one gene is necessary, one mutated copy of the gene is sufficient to cause the disease. Another condition is chondrodysplasias. Now, these are related to mutations in type 2 collagen, which is the main component of cartilage. So that leads to bone and joint deformities. okay, as you can see in these figures. And we have another syndrome known as the Ehlers-Danlos syndrome. Basically, it's a group of disorders, and they are heterogeneous. They can affect different tissues like skin, bones, blood vessels, etc. Again, just like osteogenesis imperfecta, there are different types of the disease depending on tissues that are affected and the severity of the disease. So you have some mild disease, and you have life-threatening and fatal diseases as well. And these are related to mutations in different collagen types like type 1, 3, and 5. And or mutations in the modifying enzymes like the pro-collagen and peptidase that cleaves the propeptides from the N-terminus, or the lysyl hydroxylase that hydroxylates lysines. So you can see from these images that some of the manifestations include skin fragility and extensibility. That is, you can see how skin can be extended and how joints are quite mobile. Now we have type 3 Ehlers-Danlos syndrome. It is the most important mutation. The mutation is found in the gene that synthesizes type 3 collagen, which is the major component of arteries. So that results in fragile blood vessels. Now you can see from the images the other symptoms. The other type is the other protein that is a major component of the extracellular matrix is elastin, which makes elastic fibers. It is it's a hydrophobic protein rich in proline and glycine. It contains hydroxyproline but not hydroxylysine, so it's not glycosylated like collagen. Now, formation follows sort of like similar pathway as collagen. So you have formation of the synthesis of the elastin protein, then you have hydroxylation of elastin in the ER, and then gets secreted into the outside of cells. Outside of cells, you have the formation of the tropoelastin, and then you have the elastic formation of the elastic fibers, and it is strengthened by cross-linking of lysines, just like the collagen molecule. Now, so elastin is the dominant protein, matrix protein found in arteries. It is quite elastic, so arteries can expand as blood is pumped. Something else important about the elastic fibers is that it contains cells. It prevents cells from growing and proliferating. So it maintains a certain number of cells. So any deficiency in the elastic fiber, that would result in proliferation of smooth muscle cells that surround the blood vessel. So we have a protein, or you have a sheet of tissue that surround that cover elastic fibers, and it's known as microfibrils. And these microfibrils are made from a protein known as fibrillin. And this microfibril sheet is important for maintaining the integrity of elastic fibers. So mutations in the fibrillin protein would result in a condition known as Marfan's syndrome. Okay, and that would result in weak aorta, and there are other manifestations that include a tall, thin body of the affected person, long arms, legs, fingers, and so on. Scoliosis, which is basically you have bending of the spine sideways, as you can see in here. Now, so these are the structural proteins, and I try to add a few details and information on diseases to make the topic relevant and not to make it quite only just talking about proteins and structures and stuff like that, just to make them relevant. Now, the other component is matrix polysaccharides, primarily glycosaminoglycans. We talked about these before. These are polysaccharides of repeated disaccharides. One of them is N-acetylglucosamine, and the other can be N-acetylgalactosamine. Now, the other component can be an acidic protein. So it's usually the protein is highly negatively charged, and they are modified by a sulfate group. And usually these exist as proteoglycans, meaning that they associate with proteins, forming large structures that are mainly composed of sugars and some proteins. Okay, some of them are present on the cell surface like syndicans, or they can be integrated with the plasma membrane via GPI anchors. Now, hyaluronan is one of those glycosaminoglycans, and it differs from the others in being not sulfated, and it's not part of a protein structure, so it doesn't form a proteoglycan. So the third component of the matrix is matrix adhesion proteins. Now, what these do is that they interact with collagen and proteoglycans. They link the structural proteins to each other. They link collagen with proteoglycans, and they link them as well to cells. Okay, so these are linker molecules. Now, the principal adhesion protein of connective tissues is known as fibrinolactin, which is a glycoprotein that is cross-linked to fibrils by disulfide bonds. It can bind to both collagen and glycosaminoglycans, and it can bind to cell surface proteins like integrins. Okay, so remember integrins. The other adhesion protein is known as laminin, which is predominantly found in basal lamina. So it's a major component of the basal lamina. It has a special structure that allows it to bind to integrins as well as to matrix proteins and proteoglycans. Okay. So how do cells interact with matrix proteins? How do they interact with the collagens and the fibronectins and the laminins and so on? They integrate via proteins known as integrins, and these integrins form dimers, heterodimers of alpha and beta subunits, and they can bind to short sequences of the extracellular matrix proteins and then attach cells to the matrix as well. And they can also anchor the cytoskeleton, so they mediate the interaction between the matrix proteins with the cytoskeleton, as we talked about before. I mentioned two examples of focal adhesions and hemidesmosomes. Okay, so if you remember, with the focal adhesions, you have these integrins linking the cytoskeleton, the actin cytoskeleton, with the matrix proteins. Okay, or you also have the interaction between integrins with the matrix right here, right below, and intermediate filaments, and that's in hemidesmosomes. So how do integrins do that? How do integrins link the cytoskeleton with the matrix proteins? Well, once you have interaction between integrins with the matrix proteins, you have clustering of integrins in one area and recruitment of other proteins that associate with the integrins, and these proteins link integrins to the actin cytoskeleton, in addition to a number of signaling molecules. If you remember, some of these proteins that we talked about is structural proteins, is vinculin. Okay, and you have other signaling molecules that can transmit signals inside cells, telling cells that the cell is now attached to a matrix protein. Okay, and that results in a number of behaviors in cells, such as attachment or motility or proliferation or whatever. So let's talk about cell-cell interaction now that we're done with the cell matrix. Now, there are a number of cell adhesion molecules. So there are 4 groups: selectins, integrins, immunoglobulin superfamily, and cadherins. Now, these are present on the cell surface and they allow cells to interact with each other. Okay, some of these interactions, as you will notice, are transient, meaning that they are temporary interactions, and other interactions are stable, so they last for a long time. Now, the other thing is that the interactions can be hemophilic or heterophilic. That is, interaction between 2 molecules on 2 different cells, and these 2 molecules are the same or different. So an example is selectins. Now, selectins are present in leukocytes, and these are immune cells that swim in the blood. And at the site of inflammation, these leukocytes would interact transiently, by the way, to the carbohydrates that are present on the cell surface of endothelial cells. That would then turn into stable interaction between leukocytes with the cellular adhesion molecules, which are members of the ICAM family. Okay, so the interaction again is between selectins and carbohydrates that exist on the cell surface of endothelial cells. And the other thing that is important is the stable interaction between integrins that are present on leukocytes and the intercellular adhesion molecules on the surface of endothelial cells. Then leukocytes can transverse through or between cells into the matrix of the tissue and inducing inflammation or whatever they do. Now remember that you have homophilic and heterophilic interaction. So the interaction between leukocyte selectins with the carbohydrates present on the cell surface of endothelial cells is an example of a heterophilic interaction. You also have, by the way, interaction between neural cell adhesion molecules known as NCAMs. So these are present in neurons. So you have this interaction between NCAMs with other NCAMs on the surface of a neighboring neuron. So this is an example of a homophilic interaction. And this interaction between different NCAMs is important during development. Now, cadherins is different than all of the other types of proteins that we just talked about in being predominantly forming stable homophilic interactions. So it's not transient, and it is a homophilic interaction. So you have interaction between cadherins present on neighboring cells. Now there are different types of cadherins. You have E-cadherin, which is specialized for epithelial cells, N-cadherin for neural cells, and P-cadherin, which exists in placental cells. Okay, there are different types of cadherins as well, but these are the important points. Now, what's really important as well about E-cadherin, by the way, is that this protein is present in epithelial cells, and it is important for cell-cell interaction. But when these cells turn to become tumor cells, or they can progress to cancer cells, E-cadherin expression is reduced, allowing cells to dissociate from each other, and then these cells can spread. So cadherins are important for the formation of adherens junctions. If you remember, these adherens junctions is when you have cadherin associated with proteins known as catenins, and these catenins can then associate with the actin cytoskeleton. Okay. Cadherins can also exist in desmosomes. These are the areas or regions of cell-cell interactions whereby the membrane protein is associated indirectly with the intermediate filaments. Okay. Now we can have other we can have other cell surface proteins known as desmocollin and desmogliin, but we're not going to talk much about these two proteins. So tight junctions are also important components of cell-cell interactions. Now, what these do is that they prevent passage of molecules between cells. They prevent mixing of molecules in one side of the cell with molecules of the other side of the cell, like the intestinal lumen proteins or molecules in the intestinal lumen with the connective tissue right below cells. Now, these tight junctions also separate the apical portion of cells from the basolateral side of the cell. So these tight junctions actually form a belt around cells. So it's not like they are present on just one side or one location or one small region. Rather, they form a belt. Okay. So and each strand basically is composed of a network of proteins, regardless of their names, but their names are claudins, occludins, and jams. Okay. Okay. So these are some details you don't have to worry about. Another way by which cells can communicate with each other and interact with each other is gap junctions. Now, these are channels, okay? These are channels that can form between cells. So right here you have one cell, right below it you have another cell, and you can have this interaction between cells right here via these channels. So they can exchange material with each other, okay? And these molecules can diffuse through these. Now, these channels, by the way, gap junctions, are made of proteins known as connexins where 6 of them form a connexon, and connexon of one cell can interact with connexon of another cell. Sometimes you have specialized connexons that between neurons, whereby they can transmit the electrical signal to each other. Now, there are diseases that are related to gap junctions. So these are gap junction diseases, and that includes Charcot-Marie-Tooth disease, deafness, and I know that بالمفرق deafness is widespread in certain families, and most probably it's related to mutations in the gap junction proteins, the connexins. Cataract and skin diseases are also related to gap junction defects. So collectively, we talked about a matrix proteins, and we talked about how cells can interact with each other. And with the extracellular matrix, we talked about focal adhesions involving actin. We talked about hemidesmosomes involving intermediate filaments and the important role of integrins. We talked about the cell adhesion molecules. In a number of these proteins, we talked about them, including adherins, for example. We talked about gap junctions, adhesion junctions, as well as, or adherens junctions, as well as tight junctions. So you need to know to differentiate between the different types of cell interactions.